I did something smart during this lecture, which is that I used both my normal microphone and a backup microphone in case the normal one died. It did, so you will notice a massive change in the audio during this lecture as the microphone switches. I also have bought a new microphone that I will start using in the next lecture and change the settings on my iPad so that random phone calls don't show up. Thanks.
All right. Hi, everybody. I hope that you are doing well. I am in process of grading stuff, so hopefully I'll have stuff for you on Friday, is my goal. Today we're going to start talking about aspects of T cells and MHC. And we'll also be talking about MHC on Friday. You can see that there is a problem set due after break, like more than a week after break, I think, on MHC stuff. So that's not posted yet, but it will be. This is sort of starting information for that. There also will be a paper after break that we're going to talk about. That's a pretty good paper. I'll probably be posting that before the end of this week. I think but this is sort of a sort of nice point where we're going to be starting to think about a different part of what we see in adaptive immune responses. And in thinking about what we're going to see with these adaptive immune responses and, hey, T cells and MHC, okay? I want to take a step back for a second and think about what we've learned so far and why it's not enough. Like, why do we need more immune system? What places are sort of not covered with the things that we've talked about? Because that will help us think about what things need to be covered by T cells. And so the first thing we could kind of think about is phagocytosis, because we've talked about innate immune responses, and we talked about phagocytes in terms of macrophages and neutrophils. And so here you can see an example of phagocytosis of some kind of bacterial cell getting phagocytosed and getting degraded. And you might be like, that seems pretty good. That should do the job. But it turns out that there are a bunch of microorganisms that have actually evolved ways to inhibit phagocytosis. So some of them actually get into the phagosome and have evolved proteins that puncture the phagosome and escape the phagosome and live happily ever after in the cytoplasm of the cell. Listeria monocytogenes is the classic example of this. And basically these cells are basically like, hey, thanks for letting me into a nice pH-controlled, temperature-controlled, great and safe environment that's full of nutrients. I'll happily live here in your cytoplasm. Awesome. There are other microorganisms that make proteins that actually interfere with the process of destruction in the phagosome, either by preventing that phagosome from fusing with other compartments or by just generally modifying the phagosome. So they basically make the phagosome a nice happy home for themselves and don't get degraded there. And so these are some reasons why if our whole immune response was just based on phagocytosis, it would not be good. And so you can see these microorganisms, like I said, Listeria monocytogenes, Salmonella typhimurium, and Mycobacterium tuberculosis are sort of organisms that do some of the things you see here. So there has to be more than phagocytosis. And you can say, well, of course, we already talked about more than phagocytosis. We talked about antibodies. What about antibodies? Would they be enough? And so here you can see an example of an antibody neutralizing a virus so that that virus is unable to infect a cell. However, if you look at a virus like this image of HIV, antibodies are really only able to bind to things on the outside surface of that microbe. We might see an antibody response against this yellow protein that we see here, against this red protein. Antibodies can't like cross a viral membrane or like go through layers of parts of a bacterial cell or go into the inside of a microbe. And so if we were just relying on antibodies, it would sort of be like if the airport just relied on looking at our suitcase. And we're like, that looks like a nice suitcase. You're fine, right? Because antibodies really can only interact with the outsides of microorganisms. So that seems like a little bit of a flaw. We can't see the inside of microorganisms. And in fact, many microorganisms take advantage of this by shifting parts of their outside and keeping the inside entirely the same, thus to avoid antibody responses. And so this is actually an example with HIV, but I could list so many microbes that do things like this, where you can see HIV basically just changes this outer protein. You can see it might change from purple to red. By the time we have made a good purple antibody response, well, the virus has switched to using the red version of the protein, so it can avoid those antibodies. And you can see, again, by the time we get some red antibodies, we've switched to the blue. And this is pretty easy for the virus because it only needs to change that one outside part of it. It doesn't need to change any of the other kind of structural components here. And so we see shifts like this in sort of like, you know, the virus is putting on the little glasses of mustache thing to just sort of disguise itself, but is generally not changing. And we see that kind of thing to avoid antibody responses a lot. So again, there's sort of this idea of, I don't know if I want to rely just on antibodies. The other issue that comes up that we can see here when we think about viruses has to do with viral replication. And so you can see a viral particle of HIV outside of a cell in this image. And you can imagine an antibody binding to that viral particle and potentially neutralizing it. Great. Then you can see all of the other steps in this viral life cycle. We don't need to stress about what all the steps are, but we can see that there are a bunch of steps. And the point is that all of those steps take place inside of one of our cells, in the cytoplasm of one of our cells. If I was actually to kind of put timing on this, you would see that the vast majority of this virus's life is spent inside of our cells, not as that extracellular particle. So if we were, and antibodies can't go across our cell membranes and do protection inside of our cytoplasm. They're really good for extracellular stuff and binding those extracellular particles. I will note this is the classic way the antibodies work. There is one exception, but we're not going there right now. But I know in a past year someone made lots of angry comments on YouTube that I was like neglecting that paper. So yes, I read that paper, but we're not going there right now. In any case, if we were to think about it time-wise, antibodies are only going to be able to impact this virus for a short period of its life cycle, time-wise. For many of the steps of this virus's life cycle, it's going to be basically completely invisible to antibodies because it's inside of our cells. And so you can see an awful lot of places where the immune responses that we have learned about thus far are going to be insufficient. They're not going to be good enough. We need something else. And if you look sort of broadly at all of the examples I've given you here of what the flaws are in some of these plans, what you might notice is that in general, the issue that we're seeing with all of these immune responses is that we're not able to detect insides of stuff very well. We're not detecting the inside of the virus. We're not detecting the inside of what's going on in our cells. So when things are inside our cells, we can't detect them. We think about the parts of the virus that are inside the virus, we can't detect it. Some of those microorganisms that I told you about that evade phagocytosis do that by hiding inside of our cells. And so we haven't really talked about ways that we can look and see what's going on inside of cells. We've really talked a lot about how we're dealing with more extracellular types of organisms. And so what we need to now add on to our understanding of the immune system is how exactly we look inside of cells to deal with all of the example problems that I've just shown you. Happily, the answer for all of them is the same thing. And that answer is T cells. So we have this type of cell called the T cell that is going to be able to deal with all of the scenarios I've told you about. And again, if I was sort of very big picture, very broad way of thinking about T cells, you can see, you might imagine that T cells in part have this ability to see what's going on inside of other cells and do some surveillance. So you can see all of my T cells over here on the left. Those T cells are coming from the lymphoid progenitor. So you can see we've got our hematopoietic stem cell and our lymphoid progenitor. And then, so that should tell you that these are lymphocytes. They're part of the adaptive immune response. But they are sort of coming from a different branch than B cells. So you can see that there's this B cell, or the cell is going to potentially decide to become a T cell. What you can also see here is that there are a lot of kinds of T cells. So you can see that as we get to sort of T cells, we have a lot of branches of a lot of types of T cells. Sometimes that makes it a little bit tricky to give broad definitions of T cells because some of the different kinds are really different from each other. So I'm going to give you your textbook's definition of a T cell. I find it to be one of the more useful definitions I've ever heard, but that's because it's trying to be so broad and explain all T cells. And so the textbook definition of a T cell is that T cells are lymphocytes that interact with other cells and induce them to change. So it's awfully broad, and the reason is that what they do to make the other cells change depends on what kind of T cell we're talking about. And so we're going to have to pay attention to types of T cells quite a bit as we are talking about T cells. And so when I think about T cells, one of the big classes that I divide T cells into is a class called CD4 T cells and a class called CD8 T cells. Here you can see the CD4 T cells in blue and the CD8 T cells in purple. These are distinguished, perhaps not surprisingly, using a flow cytometer based on whether they have a protein called CD4 on their surface or whether they have a protein called CD8 on their surface. And you can see CD4 and CD8 T cells doing their actions here. So the CD8 T cell, still in purple, is able to interact with an infected cell. So you can see that interaction here. And then that CD8 T cell will kill that infected cell. So CD8 T cells can also sometimes be referred to as cytotoxic T lymphocytes or cytotoxic T cells because they kill other cells. They kill other infected cells. Exactly when you're allowed to call it a cytotoxic T lymphocyte versus a CD8 T cell is something we're going to be discussing in future days. With a CD4 T cell, the idea is that the CD4 T cell is going to make some cytokines, and those cytokines are going to help other cell types act better and do their job better. And so we refer to the CD4 T cells as helper T cells because they're making cytokines to help other immune cells do their job better. And so here you can see a CD4 cell in blue producing cytokines that help this macrophage improve its killing ability. Here you can see a CD4 T cell producing some cytokines to help a B cell make antibodies better. So this is sort of the broad idea of what T cells are and T cell responses. Just like when we think about B cells, there is a unique receptor on T cells. In B cells, the unique receptor was very creatively called the B cell receptor, or the BCR. In T cells, we're going to very creatively call this receptor the T cell receptor, or the TCR. And you can see an example of a TCR on the left. You can notice that the TCR has two protein chains that are held together by a disulfide bond. Those protein chains each have a variable region and a constant region. And in a lot of ways, a T cell receptor kind of looks like an FAB structurally, just with a transmembrane domain on it. So it looks very similar to what you've seen before. You've got two chains. As we'll see later, one's going to be a heavy chain and one's going to be a light chain. You've got a variable region and a constant region. And then they have this short little transmembrane domain, a super short cytoplasmic tail. Surprise, surprise, not actually long enough to signal, so we're going to have to deal with that problem a bit later. If you recall, when we talked about antibodies, the first thing I did was introduce the general idea of an antibody / B cell receptor to you, right? And give you sort of a structure that looked like this. After I told you about the general structure of the antibody, we took a little side diversion. And we took a little side diversion to talk about what an antigen is. So we talked about the thing that binds to the antibody, or the thing that binds to the B cell receptor, for like half a class period or something. And then we came back to the B cell receptor and the B cells and what the antibodies did. We're going to do that exact same kind of overview as we think about T cells. So we are going to now spend some time thinking about what the T cell receptor binds. When we did this with antigens and B cells, that took me about half class. When we talk about what the T cell receptor binds, it's going to take me about 4 classes. And so it's actually the same process. The same, maybe 3, I don't know, 2.5. It's going to take me a little while. It's the same idea. I'm basically now switching us to our little talk about the antigen before we come back to the T cell and its receptor. It's just the antigen's a bit more complicated to talk about, okay? Because if you think about it, what I've told you so far about T cells is that T cells have this ability to see inside of other cells. Like, do T cells have X-ray vision? Is there receptor X-ray glasses? Like, you know, it sort of gets a little tricky of like, what exactly is this receptor seeing if it's seeing inside of cells? And there are a couple of different chains of experiments that people have done that really gave us a lot of information about this. One of those is related to the Zinkernagle and Doherty experiment, which is like such a classic experiment in immunology, we have to talk about it. And it really helped us to answer this question about what's going on with T cells. And introduced us to some key things about T cells. Like many experiments that I go through in class, This one won the Nobel Prize. I believe this was the 96 Nobel Prize. And really kind of defined some of the things that we're going to be talking about thus far. And so Zinkernagel and Doherty had mice of two different strains. One they call strain A. The other they call strain B. Every mouse who is strain A is genetically identical to each other. Every mouse who's strain B is genetically identical to each other. There's some genetic difference between strain A and strain B. Zinkernagel and Doherty don't know what it is at this point. Again, as I will tell you much more about later in this semester, so I'm going to tell you this now as like a caveat, like a little asterisk, and you are welcome to forget it until much later in the semester when I'm going to be like, oh wait, remember that thing. So it's okay if you don't remember this, but what I will tell you is that this experiment came out very cleanly because, again, they made some lucky choices in terms of the types of mice they used. There is another complication that could come up here that we're going to deal with later in the semester, which they could have seen if they happened to have chosen different mice. But they didn't. They chose the mice they chose. So they got the nice clean answer. So yes, I'm giving you the clean answer in this right now. And later in the semester, we need to see a slightly messier answer. And so what Zinkernagel and Doherty did is they took a mouse, and you can see they started with a mouse of strain A, and they infected that mouse with a virus. And that's a virus that you can see here is called LCMV. You don't actually care that it's LCMV. And they got some, and they sort of let that mouse make a T cell response to LCMV. So they infected their strain A mouse with this virus, LCMV, and then they got the T cells out of that mouse about a week later. And these were T cells that could respond to LCMV. These were sort of the activated T cells that started doing something about LCMV. And what they wanted to do is they wanted to look at the activity of those T cells. The T cells that they used in this experiment were CD8 T cells. And so they were looking for the ability of these cells to kill other cells. And so the basic idea is that they put these T cells into a dish with some other type of cell. And their goal was to determine if this other type of cell got killed. We do these assays pretty frequently in immunology. I guess I will give you my slight caveat or slight disclaimer here. I did my PhD on CD8 T cells and viruses. And so like when I just think of an example in my head, it's always a CD8 example most of the time. And so like I've done experiments like this. This feels very much like home to me, thinking about these types of experiments. And so what they were trying to do when they do this experiment is they're doing this assay to see, can my CD8 T cell do killing? So they're looking at something called a cytotoxicity assay. So they're looking at their CD8, they want to see if their CD8 T cell can kill some other cell type. And the way that they did their cytotoxicity assay is shown here. We still sometimes do assays like this, although we have some modifications that are a little safer now. But this is, like I have done this assay. You start with the cells that you are thinking are potentially going to get killed. They're called the target cells. So you start with your target cells. And you take those target cells and you load those target cells with radioactivity. Specifically, you use radioactive chromium in this case. So you're going to label those cells with radioactive chromium. So you're going to have radioactive chromium in the cytoplasm. You're then going to wash away the excess chromium. So here you can see the cell has just chromium inside. Then you're going to allow the CD8 T cell, you're going to put the CD8 T cell with the target cell with the idea of allowing that CD8 T cell to potentially kill that target cell. And what you're going to then measure is how much radioactive chromium or how much radioactivity gets into the supernatant. how much radioactivity comes outside of cells. Because if our target cell is killed, it's going to lyse and it's going to lose the radioactivity and put it in this media. So this is often referred to, officially this is a cytotoxicity assay. A lot of times people talk about this as a chromium release assay because you're looking to see if the radioactive chromium got released. So Zinkernagel and Doherty made a bunch of target cells with chromium. They got those CD8 T cells from a mouse of strain A that had been infected with LCMV, and they were looking at the ability of those CD8 T cells to kill the target cells. And when they did this, they used a couple different kinds of target cells. I'm going to talk through a few different options of target cells. The pictures show some of them. I'm going to talk about a few other ones just to kind of make this, to get some breath. So one of the things, so what they use for two of their types of target cells are shown here. One type is also from a strain A mouse. So it is a mouse that is genetically identical to this mouse that they used originally. These are just some cells from that mouse that have been infected with LCMV. So these are like infected cells of strain A. They also have some cells that are infected cells of strain B. So we've got our strain B infected. I'm going to actually list out 4 kinds of cells here as target cells that they looked at in different types of experiments. So one of them would be LCMV infected cells from strain A. One would be LCMV infected cells from strain B. And remember, strain B is genetically different from strain A. One uninfected would be uninfected cells from strain A. So strain A cells that don't have the virus, normal strain A cells. And one would be strain A cells that were infected with a different virus. So perhaps influenza infected strain A cells. So these are our different types of targets. And if you remember, they started with CTL from a strain A mouse that are recognizing LCMV because they infected that mouse with LCMV. Sometimes when we're thinking about this, we refer to these as the effector cells. So they mix together effector cells with this target, or with this target, or with this target, or with this target. They did sort of 4 different experimental dishes of this. And in each case, they looked at chromium release cytotoxicity. And they saw that these CTLs only were able to kill in some situations. So the CTLs were only able to sort of do their job sometimes. So if we had a situation where the target had been infected with the same virus, LCMV, and it was the same genetic type, strain A, then we saw the CTL killing. The T cells could do their job. And that's also shown up here, strain A, LCMV infected. If they used target cells from strain A that were uninfected, that had not been infected with the virus, the CTLs, the cytotoxic T cells, didn't work. They didn't kill the cells. So somehow the cytotoxic T cells knew which cells were infected and which ones weren't. And they only killed infected cells. It's not shown here, but if we looked at target cells that had been infected with influenza from strain A, we also did not see the CTLs killing. So the CTLs didn't just know infected versus uninfected, they knew ones that were infected with the virus they'd seen before. So this is another example in the adaptive immune response of specificity. Those CTLs are specific only for the virus they were activated against. They're not killing any virus-infected cell. But the really key thing that Zinkernagel and Doherty found and kind of won the Nobel Prize for is in this strain B situation. So here in strain B, so we've got target cells from a different genetic background, different genetics than the effector cells. And now those effector cells don't work. So if the T cells come across an infected cell from a different genetic background, they don't kill. The idea here is that T cells seem to need to have a couple boxes get checked. They need to both know that something is the right pathogen, but they also need to know that they're looking at the right kind of cell, a cell that is theirs, a cell that is self. So there's this recognition, not just of the microbe, but of some aspect of your genetic makeup too. Because these effector cells couldn't recognize targets of the wrong genetic makeup. They could only recognize targets of the correct genetic makeup. You can kind of think about this that the target cells were restricted in who they were allowed to act on. What we now know is what this self gene, self protein that's encoded by the self gene is. So there is this protein called MHC. It is the self protein, and it is something that the T cell receptor is seeing in addition to seeing what's coming from the pathogen. So if you look at our T cell receptor, we've got our part of our pathogen in red, we've got our MHC in yellow, and what you can see is that the T cell receptor is actually binding to this combination of MHC plus peptide. So the T cell receptor is sort of saying, is the MHC right? Is the peptide right? So in this case, the peptide would be from LCMV, it's right. The MHC would be from strain A. Yes, yes. Here, maybe yes for the peptide, no for the MHC. Here, no for the peptide, yes for the MHC. Here, no for the peptide, yes for the MHC. So sort of there's this extra recognition of this self protein. And what you can also see is that this MHC protein that is shown here is on the surface of cells, and it's taking some material from the inside of the cell. So you can see we've got some material in this red stuff that's inside the cell, and then it's going to end up with the MHC outside of the cell. And so sometimes I think about, I've seen a lot of different analogies for MHC. Sometimes I think about it as like a little, I think Miller in her non-majors class says that it is like a little toddler who got a trophy. And it's like, mommy, look what I got. I like to think about it a little bit more like, it's just a flagpole, or it's just some kind of thing where you're like, this is what I have. Am I okay or not? Here are the things inside of me. Tell me if I'm okay or not. And so I think of the MHC as like the flagpole, or like the pedestal, or the shelf, or whatever that you're putting stuff on to show it off and to see if whatever it is okay. And so you can see the T cell receptor is checking out the MHC and is checking out the peptide. And so the thing that Zinkernagel and Doherty really discovered was this phenomenon known as MHC restriction. So there is this protein, MHC. You can see the examples of MHC either in yellow or green here. You can also see the little peptide that's coming from the microbe. And you can see that the T cell receptor is looking at both the MHC and the peptide. If it's the right peptide but the wrong MHC, the T cell doesn't work. If it's the right MHC, wrong peptide, T cell doesn't work. So that your T cells are restricted to only act against your MHC. And again, you can see sort of a general overview of this process here. Again, in my super oversimplification for the moment, I will just tell you that this MHC recognition and the details about MHC recognition are the reason we do transplant matching. When we're trying to do transplant matching, we're matching MHCs. That's what we're trying to match. And the issues with MHC restriction are why the transplants don't work. We're going to specifically talk about transplantation immunology, where I do have to kind of tell you some more complicated parts to the story much later in the semester. Paige, you have a question?
Do we only have one type of MHC?
That's where we're going. Great job previewing what I'm going to start talking about. Other questions? So Paige asked a question about, do we have only one type of MHC? And that's really key to think about. There's also one other piece of this that's really key to think about, in the way that I've kind of been implying things about MHC, which is that I implied that MHC is different. I talked about your MHC versus a different MHC. You know, strain A mice and strain B mice have different MHC. So I've kind of also implied that everybody's MHC is different. And so we sort of have to get at these issues of like MHC variation here. But before I tell you about MHC variation, I want to tell you one other thing about MHC and its general structure. So what you can see in this image is that we have some protein from our pathogen in the surface of the cell. You can see it gets degraded, gets cut up in some way, and then ends up getting put in this MHC molecule. I will often talk about the pocket or the cleft. It's sort of put in a little pocket in the MHC molecule to get shown off, officially known as the cleft. That little piece of the pathogen, that red dot, is really what we think of as the epitope here. So this whole protein is the antigen, and that small piece that makes it into the MHC pocket is the epitope. When we talked about antigens and epitopes with B cells and antibodies, we did not care about your personal genetics. In fact, if I tried to get the antibody against SARS-CoV-2 from all of you, it would be identical. Because it has nothing to do with you as a person. It's about the structure of SARS-CoV-2 and what the epitope is. But we also talked about the fact that the antigen, in the case of B cells, might be a protein, might be a carbohydrate, might be a lipid, or might be a nucleic acid. I told you it could be any of those things. With T cells, the epitope is only things that can get into that pocket, only things that are compatible with that pocket to be shown. And there's only one type of thing that can get into that pocket, which is a peptide. So MHC only can show peptide antigens. T cells only respond to proteins or to peptide antigens. You can't get T cells responding to lipids, carbohydrates, nucleic acids, because none of those can make it onto the MHC molecule. Further, if you remember, we talked about how the epitopes with antibodies and B cells are in the three-dimensional folded conformation of the molecule, and you could denature it and you could ruin things. It might just be, some loops that are only next to each other when you fold the protein. With MHC, the protein gets degraded before it gets presented on the MHC molecule. And so you are always seeing a linear stretch of amino acids from basically the primary sequence of the protein. So you're always getting a linear piece. You're not going to have an epitope that's this loop, and this loop. You might have that for T cells, but for MHC, you're going to actually take this protein apart, and only a little string of amino acids that are next to each other in the peptide can potentially be an epitope. So with MHC, we're always presenting linear epitopes. So a linear stretch of amino acids from the protein, and we're only presenting peptides. And what you can notice is you're going to see this image so many times over the next few lectures. I don't know, like this image just makes a lot of sense to me, so I come back to this a lot. So you can see my MHC molecule in green. You can see that it's got a little pocket that has a specific shape. And you can see the peptide in brown that's going to be bound in that pocket. And you can see that some unique features of that peptide might be involved in whether or not you're getting binding. We're going to talk way more about that going forward. And you can also see that some other parts of that peptide are going to be present for the T cell receptor. The T cell receptor is making contacts with the peptide and with the MHC molecule. But specifically, the reason why I show you this here is so you can kind of see that pocket or see that cleft. Okay, because I'm going to be talking about that pocket or that cleft. And what you could imagine in thinking about different MHC molecules, if they existed, is that if they had a different pocket, they could show a different peptide, right? They could put different peptides in pockets of different shapes. You can see this here as well. So here at the bottom, you're actually seeing a structural view of MHC. And in yellow, you can see the peptide. So that peptide is really kind of in a little pocket. In some information that's really going to be Friday material, I'm going to tell you about one aspect of this. And in some immunology textbooks, at this point, they actually show a photograph of a hot dog in a hot dog bun, as if you don't know what that looks like. So like, when we're talking about a peptide in this pocket, you can sort of have that kind of imagination. And here's the T cell receptor interacting with that MHC and that peptide. You'll notice the part of the T cell receptor that's interacting with MHC plus peptide is the variable, or V regions. And specifically, if you look at which parts of the V regions are touching the MHC plus peptide, it's these little loops. It's 3 little loops from each of the 2 chains interacting with the MHC plus peptide, just like you saw with the antibody and the B cell. So again, it's those same 3 little loops. Here, you can see that overview of a TCR and an MHC plus peptide. And here, you are the T cell, and you are looking down at the MHC plus peptide. This gray blob is the MHC, and this yellow is the peptide. And so you can see it's sort of this contiguous surface that you are seeing as the T cell receptor of this peptide kind of in a pocket of the MHC. You can even see the places where my 3 little loops, loop 1, loop 2, loop 3, loop 1, loop 2, loop 3, are actually making the contacts and touching. So it's, again, very similar to what you've seen with the B cell and the antibody. We even know a lot about the specific angle that the MHC has to be coming at to bind the T cell receptor. And so this is actually showing you the T cell receptor's angle across the MHC in both top and bottom. So we understand a lot of the details of these interactions. But now we've got to get sort of to Paige's question and to my general idea about MHC diversity. And so we're going to think a little bit about MHC genetics. I'm going to say much more about MHC genetics on Friday. I'm only going to give you a very high level definition of a couple things now. We're going to zoom more in on Friday. But MHC is found on the short arm of chromosome 6 in humans. And it's basically the entire short arm of chromosome 6. It's a big part of your genome. I can always remember that because there is a author, and now I can't remember the name of the author. I sometimes say Michael Crichton, but it's not Michael Crichton, it's somebody else, who wrote a book called Chromosome 6. That's why I can always remember this is chromosome 6. And in the book, the idea is that they give primates, they give certain monkeys human chromosome 6 because they're trying to do fun transplant immunology, and they make them like sentient. And it's this whole like crazy sci-fi book. But I can always remember chromosome 6 because it's in this book, chromosome 6. The MHC genetic region is pretty big. We'll go through specific parts of it, like I said, on Friday. But right now what I want you to know is that there are different parts of the MHC locus. There's a part called the class 1 region, there's a part called the class 2 region, and a part called the class 3 region. The class 1 region encodes some genes that are called the class 1 MHC molecules. And so I'm going to talk about class 1 or MHC class 1 a lot. The class 2 region encodes things that are called class 2 MHC molecules. And so again, today I want to mention some things about class 2 a few times. So I'm going to be talking about class 1 and class 2 a bit. So you need to know what those are, just types of MHC. They're encoded in different parts of the chromosome. We're going to see much more about them on Friday. There is also a class 3 region. The class 3 region actually does not encode MHC genes. But weirdly enough, that part in the middle of the chromosome is a whole bunch of other genes that are really important in the immune system. So it's like that whole section of your chromosome is all immunology stuff. Class 1, class 2, and like other useful things like complement and like some cytokines and things like that. So that's what's in the class 3 region. But really what you need to know right now is that there's MHC class 1 and there's MHC class 2. In humans, when we look at an individual chromosome 6, we find that we have 3 kinds of MHC class 1 on your chromosome 6. One of those types of class 1 molecules is called A. Really is called HLA-A. One of them is called HLA-B, and one of them is called HLA-C. We also can look at our chromosome 6 and look at our class 2s in humans. And we also have 3 class 2s. So we have HLA-DP, HLA-DQ, and HLA-DR. We will see the nomenclature in mice on Friday. What I really, so note that there's 3 class 1s, 3 class 2s. The class 1s have 1 letter. The class 2s have 2 letters. So A, B, C, DP, DQ, DR. So that is one thing that might help you recognize them. So to start to answer Page's question about are there different MHC molecules, you've got 3 class 1s, HLA-A, HLA-B, HLA-C, and 3 class 2s, DP, DQ, DR on chromosome 6. I will also tell you that because I always do, I often use just class 1 with my examples, because I've spent a lot of my life thinking about class 1. So you'll see class 1 come up as an example in a couple places. But you have 2 chromosome 2s, or chromosome 6s. You also have 2 chromosome 2s, but that's not relevant here. You've got 2 chromosome 6s, one from your mom, one from your dad. Which means that you have an HLA-A from mom and an HLA-A from HLA-B from mom and HLA-B from dad. An HLA-C from mom and HLA-C from dad. A DP from each, DQ from each, DR from each. And what's really interesting about these molecules is that these molecules are codominantly expressed. What that means is that you make all the proteins. It's not that you make one or the other, you make them both. So if you look at the surface of one of your cells, you can see that you would have two HLA-As. Here I show it as A1 and A2, A1 coming from mom and A2 coming from dad. You've got two HLA-Bs, B1 and B2. You've got two HLA-Cs. And you've got two DPs, two DQs, two DRs. So you've actually got 6 class 1s and 6 class 2s on the surface of the cell. If you look at all of those different MHC molecules, big picture, class 1 and class 2 have different structures and do lots of different things. But if you look at the 6 class 1s or the 6 class 2s, the place where they differ from each other, here's what is in the pocket. So if we actually look at different MHCs and we try to figure out where are they different from each other, it's in this red spot. It's in the pocket. And so the idea is that these different MHCs are going to bind to different peptides. So the idea is you have 6 class 1s on the surface of all your cells. So you basically have 6 pocket shapes on the surface of your cell, giving you 6 choices or 6 chances to show off parts of the pathogen. So you got 6 different sort of pieces of a pathogen you can show off, because you've got 6 class 1 pockets. You got 6 class 2 pockets, you show off 6 different parts of the pathogen, because these are going to differ from each other based on different pockets. Does that make sense? Okay. So if you think about it, this is like best case scenario here. Mom and dad each had a different A, they each had a different B, they each had a different C, they each had a different DP, they each had a different DQ, they each had a different DR. So you have the full 6. 6 different class 1s, 6 different class 2s. Okay? So now what we have to think a little bit about is that actually the scenario we see in real life? And before we think about this, I want to think about a different gene for a second, okay? And again, we're doing this in a somewhat simplified fashion, but I think it gets us where we need to go. So let's imagine that I looked at your 2 copies of actin. The actin you got from your mom and the actin you got from your dad. Okay? Do you think the 2 actin copies that you got from your mom and from your dad are the same or different from each other? What do you think?
I think they should be the same because they're very essential.
Yeah, they're the same. You hope you have 2 normal old actins that are making the same actin that's like good normal actin, right? You don't want to like mess with actin. So you probably have 2 normal actins. What would happen if you had like a not normal actin? You probably wouldn't be very good at having cells, right? So you probably have 2 good actins, right? Let's imagine we looked at everybody in this whole room's actins. So there's 20 people in this room. We each have 2 actins, so there's 40 actins in this room. Do you think we have 40 of the same actin, or do you think there's variation? And if there's variation, is it a lot or low? Yeah, there's basically no variation. We'll say no variation, yes. But there's really no variation, especially at the level of the protein that you make. We pretty much all make identical actin. And it's really important that we do that because this is such an important gene that we do not want variation. Somebody has a mutation in their actin, they're probably not going to survive. For most genes in the human genome, there aren't a lot of versions. We usually say there aren't a lot of alleles because we want everybody to have the same good one. Okay? Here, I'm assuming that mom and dad have different versions, have different alleles, which is different than what we're saying with actin, right? This is sort of actually a unique thing that I'm telling you that mom and dads are definitely going to be so different. The reason why I'm saying this is because if we actually look at the MHC genes, they are the most polymorphic genes in the human genome. What that means is that there are the largest number of alleles of these genes out of any gene in the human genome. These are the genes that vary the most in the entire human genome. So in our example, we said there was one actin in humans, right? For HLA-A, there are 2,980 different versions. And honestly, this textbook was made, I think this is like a 2020-ish textbook, I can't remember. People are discovering new ones all the time. There is actually a website where you can get the most up-to-date number. I don't have it off the top of my head, but every number on here is too low because we've discovered more. So this says 2,980 HLA-As. There are 3,700 HLA-Bs, 2,661 HLA-Cs. DMs, you can see it's only like 11. DQ is 838 plus 35. DP is 695 plus 32. So what you can see is that this gene is really unique compared to a lot of other genes in that we haven't had natural selection pushing it so that we all have the same one. Natural selection here has actually worked the opposite way. It's good for this human population to have mutations in MHC and to have as much variation as possible. We actually really want a lot of variation in MHC. And as a result, we have the most polymorphic gene in the human genome as MHC, the one that has the most different types of alleles. The idea here is that each of these different, that you really should, hopefully, be in this situation where mom and dad are giving you different A and different B and different C, and you're really getting 6 different class 1s on all your cells and 6 different class 2s. And why is that a good thing for you as an individual? Yeah, Roxy.
Because then you're able to call back 6 different things, like 12 different stuff altogether versus just
Okay.
6, like.
Okay, so why is that important? Let's sort of like phrase it in another way. You're totally right, but let's look at phrasing it.
Well, if everything's self-recognizing and each person has their own kind of unique thing, then if something were to happen, if my MHC was in Carissa, then her cells wouldn't recognize it.
Okay, so we're gonna get to that. That's one issue. Yeah, I don't know.
It's just like, what if you got infected with a virus and then you don't have a pocket for it?
Yeah. What if you got infected with a virus and you don't have a pocket that can show off a piece of that virus? That would be really bad, right? The virus could basically hide from T cells. You don't want that. You would die of that virus. So what's great is that you want to have 6 choices of class 1s. You got 6, 1 out of 6 of them might work. You got 6 class 2s, 1 out of 6 of them might work. If you didn't have that kind of variation, if you only had 1 MHC class 1, you only have 1 chance, and it would be really easy for a virus to get to a situation where it could just hide from your pocket and hide from T cells and not be bound by its pocket. And so this is sort of part of why MHC diversity is so important. It's really important that you have really, really diverse MHC. Yep, Jazz.
How likely is it that you inherit the same HLA from mom and dad?
Well, so there's 2,980 of them. I will say, and this comes up a little bit later, they're not all equally frequent, and so it's sort of the question of if they happen to have the really frequent ones or if they have really unfrequent ones. But we're going in that sort of direction.
Thank you.
Yeah. And so this is just, these slides are sort of showing pictures of basically what I've been telling you. So here you can actually see, this is the pocket. It has spots for particular parts of the peptide, and by having diversity in the MHC, you're going to have sort of 6 different pocket shapes that can accommodate 6 different types of peptides. If we're in the case of class 1, 6 different types of pockets, 6 different peptides in the case of class 2. This is going to be something that we spend much more time on Friday, so I'm just going to sort of blow through this right now. But for those of you who sort of think in the way of kind of more biochemical things, the way that this actually works is that the pockets accommodate certain amino acids. And so, for example, this pocket that's listed as KB can fit any peptide that is 8 amino acids long, 8 or 9, that has a phenylalanine or a tyrosine there, because of the shape of the pocket and what kind of like bulge it can hold. It can hold an R group of a phenylalanine or a tyrosine, and it usually wants a lysine or leucine or an isoleucine at position 8. And then you can see a different pocket will have sort of a different preference. So here it wants a tyrosine at position 2. But what you can also notice is that you can have many peptides from many different microbes that all kind of meet that criteria. And so one pocket can give you information from a lot of different microbes, all of whom might have a peptide that sort of matches the criteria for that pocket. But we'll see much more of this on Friday. So, we talked about this a little bit in terms of why it's good for you to have kind of diverse MHC, right? What I also want to point out when we think about different MHC molecules, they're all presenting different pieces of the microbe. So one of them might be showing off the very N-terminal piece of some protein. Some might be showing off a middle piece of the protein. Some might be showing off a very C-terminal piece. Each in a sort of whichever one matches the pocket that you have. As a result, each of those are going to be turning on a slightly different T cell, because the T cell is seeing that peptide along with the MHC. So you're going to get a different T cell in different people. If I and Julie were infected with the same microbe, our T cells would actually be different T cells that were responding. With antibodies, it would be identical antibodies. With T cells, they're different, because it's dependent on our MHCs, both what's the structure of our MHC like, what's the pocket like, what peptide can it show. All T cell responses are not equally good. Some are better than others. And so it turns out that if we look at different MHC types in people, we see that they have a different outcome in response to different infections. So here you're looking at HIV, and we've got a list of a bunch of different MHC types. And so you can see, for example, if a person has HLA-B58, they have a high amount of virus and they have sort of rapid disease. The idea is that that HLA type is bad for HIV. If you have HIV and you have that HLA type, you're going to have worse disease. If you have a different kind of HLA, for example, B27 is a particularly famous one, you're going to have really low amounts of virus and you're not going to get very sick. And so we see big differences in the human population in terms of whether you have a HLA that helps you make a really good response against a particular microbe or a really bad response. This is one of the big reasons why we see such variation when everyone is infected with the same microbe. One of the reasons why we see a lot of variation is because everyone is making a different T cell response with their MHCs. You might say, hey, I don't like this business. I would like the good MHC. And the trick is there's no one good MHC. Different MHCs are good in one situation and bad in one situation. So whatever MHC you have, there are some microbes where you're going to fight them really well, and some where you're going to fight really badly, based sort of on probability. And there are, for example, you can see B27 is one that has been described as being really, really good for HIV. Oh, yeah, they're coded. I know it in my head. B27 also is the most common MHC type to have the autoimmune disease ankylosing spondylitis. And so if you have B27, you're going to be good with HIV, but you might get this one autoimmune disease. And so every different MHC type has sort of pros and cons. There's no one that's good for everything or no one that's bad for everything. And this is why it's really good for us as a human in the human population to have such variable MHC. Because the idea is there's no pathogen that can kill us all. Some pathogen might evolve and it might suck for me with my MHC. But somebody is going to have the right combo of pockets to deal with that MHC. And so this is why natural selection has sort of selected for breadth or diversity. This is known as diversifying selection. Instead of selecting for just one different type of allele. So with the rest, so basically I'm going to spend the rest of the time kind of doing some fun examples. I don't know what order I have my slides in at this point, but I do want to tell you about some fun examples. So one fun example gets sort of to this. So as you are now thinking about your MHC, you're imagining that you're going to go home and sequence all your MHCs. Yes, I have priced how much it would cost to do that, either commercially or myself. No, I have not done it. But if you're going to go, so what do you hope you find in your MHCs? What do you really, really hope is the case if you look at your MHCs, thinking about this? Yeah.
12 different types.
You hope they're all different. You hope mom and dad did not give you any of the same ones, like Jazz said, right? And so if you think about it, it might also, if you think about your kids, ideally you want your kids to have really diverse ones, right? You want your partner to have really different MHCs than you. In a perfect world, right? People have actually tried to do studies on this to see if we are attracted to people with MHCs that are similar or different than ours. I think these studies are nuts. However, people actually did this study. If you're going to start to ask me some of the mechanisms behind this, I can't tell you it. I could handmade one if I really had to. But this is actually known as the sweaty t-shirt experiment. And so what they did is they took a bunch of female college students and a bunch of male college students, and they had the male college students work out. And then they gave the female college students the sweaty t-shirts of the male college students and asked them which ones smelled the best. Which one were they most attracted to based on smell? Yes.
Was it like, did it smell like good or just like not as bad?
It depends what
MHC
MHC.
I don't know. I don't know.
Alright.
But in any case,
That doesn't smell as bad. Maybe you did do a sweat.
Sweat is sweat.
the ones that, people found most attractive were the ones that were the most distinct MHC from them. So there seems to be some weird thing about attraction to people with really different MHCs than you. I have to hand wave a lot before I can even come up with some kind of mechanism. So no, I don't have an answer. Though when I used to teach this class in the spring, this often fell right around Valentine's Day. And I would be like, if there's that person and you just can't figure out what it is about them, maybe it's their MHC.
So what, is this like evolutionary, attribution to the fact that like you don't want to reproduce with like family members who potentially have similar MHCs to you?
Evolutionarily, yes, that makes sense. How the heck you are smelling someone else's MHC? I have no idea.
Oh, you know.
Are pheromones coded on chromosome 6?
No, pheromones aren't necessarily gene. If I had to make a mechanism, I would say that your MHC influences your microbiome, and that what you're smelling is the microbiome.
You know about the microbiome.
But that's me having to like try to make an answer. No one's actually shown any of that. Okay? In addition, this again sort of gets at some of Jazz's questions. There are big differences around the world in frequencies of MHC types. And so if we actually look at different populations, we see different MHC types as being more or less similar. And so this actually becomes a really big deal when we think about like transplantation, because in fact sometimes there is a lot of requirement for transplantation for similarities in ethnicities, and it's not because of like people being weirdly racist about what kind of kidney they want. It's because in fact there is sort of differences in frequencies of MHC genes in different groups. And so we can actually see different populations very much having different MHC types. Anthropologists have used this information. So there, for example, are a bunch of Pacific peoples, and it was unclear where they came from. Did they come from North America to the Pacific, or did they come from Asia to the Pacific? And it was figured out by figuring out whose MHC they had and where they had the closest MHC type. So MHC is used in a lot of different settings. This also means that when we think about treatments of diseases, when we think about vaccines, we actually have to take into account where we're going with our treatment, because the MHC types are going to be different and we might need to do some different immunology. Here's my thing that I thought I had before about disease associations. The moral of the story is lots of diseases are associated with MHC, and it's all different. Sorry, I want to talk about the cool ones. But what I want to spend the rest of the time talking about is, what if MHC is not diverse? So what would happen if MHC was not diverse? And I want to give you 2 studies of 2 different organisms where MHC is not diverse. One of those organisms is the cheetah. So what do you guys know about cheetahs? Just like general things about cheetahs. Okay, they run really fast. Yes, correct, yeah.
Aren't they like all identical?
So there's something about all identical. What else do you know about cheetahs? In general. Okay, besides again them being really fast.
That's always vicious.
They're also super endangered.
Oh yeah, true.
They're super, super, super endangered. The reason why they're super endangered is because they're all basically genetically identical. And as a result, they all have the same MHC. And so if a pathogen comes around that can avoid the cheetah MHC type, then it's going to kill off the remaining cheetahs. And so when you do things like go to cheetah rescue, you have to spend a lot of time disinfecting hands and feet and all sorts of things before you're allowed to pet the cheetahs. Because we really have to pay a lot of attention about making sure that the cheetahs are not getting different microbes. So cheetahs, one example of what would be bad if our MHC was not diverse. So yay, we are not the cheetahs. But then there's the other example. This organism, if you are not aware of it, is called the Tasmanian devil. I love this image of the Tasmanian devil the best, because when you look at it, you learn a lot of key things about Tasmanian devils that you need to know for this story. If you Google Tasmanian devils and read their Wikipedia page, you will find out that a large portion of Tasmanian devils' life and history and everything involves biting. And you can see this guy is going to bite you. Tasmanian devils interact a lot by biting. When Tasmanian devil wants to interact with another Tasmanian devil, it bites it. Sometimes in the face. Tasmanian devils have really similar MHC. Basically, they also all have the same MHC. They all died off in the past. There were like a couple progenitors that gave us the rest of the Tasmanian devils we have. So they all have the same MHC. If right now I had skin cancer on my hand and I wiped it on Aurora, Aurora would not get skin cancer. And you want to know why Aurora would not get skin cancer? Because our MHCs are different. It would actually be like a transplant that she's rejecting. The way she would also reject a kidney that came from me. Okay? Tasmanian devils all have the same, so cancer, unless it's like a viral cancer, is not transmissible, right? Tasmanian devils all have the same MHC. They all spend their time biting themselves and biting each other in the face. And Tasmanian devils are probably going to go extinct because of the Tasmanian devil facial tumor, which is a transmissible cancer. The only one of only two transmissible cancers really well understood that is able to transmit from face skin to face skin of Tasmanian devils because they have all the same MHC. And like I said, this is probably going to make Tasmanian devils go extinct. In fact, there has been a more recent genital cancer in some wild dogs that seems to be transmitting in the same way. And now we actually have realized that there's two different strains of Tasmanian devil face tumor that are probably going to make them extinct. So hopefully you got some fun facts with Tasmanian devils and cheetahs and sweaty t-shirts. And we're going to talk more about the structure, sort of the biochemistry, cell biology of MHCs on Friday. I will see you guys then.